So do we have Leucadia? Is that Doug from Leucadia next? I know that we've had to juggle a little bit because of a technical glitch in the order.
Great. Well, thank you for inviting me to this very interesting conference. I've enjoyed the last few days, and it's great to have the last word. So I'll share my screen. Let's see, there we go. Can you see?
Mm-hmm.
Okay, great.
It's good.
So Alzheimer's disease is the holy grail of healthcare. 40 million patients worldwide, and there's no cure. This area has become a graveyard for big pharma, with $30 billion invested in an unbroken string of failed clinical trials, including Biogen's. At Leucadia, we've uncovered an anatomical trigger for Alzheimer's disease that's let us develop a non-invasive method to predict who will get Alzheimer's disease and when, years before they have cognitive decline. We've also developed, oops, wait a second. There we go. We've also developed an implantable device that fixes this root cause because it's an anatomical issue, surprisingly. So today I'm going to talk about the research that's led us to these conclusions and the innovative products that have come from it. So Alzheimer's disease causes atrophy of the brain, as you can see here on the right-hand side. And that's due to the loss of billions and billions of neurons. Now, it affects the entire cerebral cortex, but it doesn't start everywhere. It starts in a very specific spot. As you can see here on the right where it says early AD, that orange area above is the middle of the brain sort of cut in two and opened up. It's called the medial temporal lobe. And if you look below that, that's the underside of the brain. There's one on each side. So it starts there and then it spreads outward as the disease progresses, eventually to the entire cerebral cortex. It's a bit like a campfire that turns into a California wildfire. Okay, the question is, why does it start there? It always starts there. What is peculiar about that part of the human brain that predisposes it to disease? And can we stop it? Well, yes, it is, because it's really a plumbing problem. Okay, here we have a schematic of some cells in the body. The circles are cells. Okay, all cells, they make two types of waste products, basically. There's small ones and large ones. An easy way to think of it might be for New York. Okay, think of all these brownstone buildings as neurons in a brain. So people in the brownstone produce small waste, say a dead goldfish or something else in a toilet, right? And it gets flushed away through the system. If you look above in the cells, cells make carbon dioxide. It goes into the bloodstream and gets carried away. All right. They also make larger trash and debris, trash and the odd sofa, and it has to get carried away by going in between the cells. And that also happens in the body. If you look above, you have interstitial fluid that flows between the cells and picks up debris, insoluble macromolecules, pieces of dead cells, and it carries it to the lymph nodes. Now, this is done by interstitial fluid that starts in the blood vessels. There are tiny holes in the capillaries where plasma comes out to create that. But that doesn't work in the brain, although the brain still needs to clear those things. To do that, it makes something called cerebrospinal fluid in 4 ventricles or cavities or chambers in the middle of the brain, shown here in blue. So the CSF, about half a liter a day, is produced there, and it slowly migrates out through the cells, picking up detritus and carrying it to the surface of the brain where the subarachnoid space is. Okay, and there it leaves the brain. The area where Alzheimer's disease starts is shown here in red in this. It gets cleared out. It makes its way to the surface and to the front of the temporal lobe, and then it goes out through the olfactory system. What do you say? Well, let's look at this brain from underneath. Okay, at the bottom, the orange area is the medial temporal lobe where Alzheimer's really takes off. Okay, so the debris is carried to the outside of that, and then it moves forward into a rudimentary funnel formed by the lateral olfactory stria. And it moves along that past the surface of the basal forebrain, another early area that's hit in Alzheimer's disease, and carries it to the olfactory bulbs, okay? And from there, it leaves the brain through the skull. This is how it does it. So here's a skull. If we take the top off and we're looking at it from above, the top of the image is the front. So those two little depressions there are the cribriform plate, which looks like this, okay? The olfactory bulbs sit like two little sausages in the cribriform plate. And you notice that the cribriform plate has holes in it, and that accommodates bundles of sensory fibers carrying smell senses of lunch or the dog up into the olfactory bulb. And around those bundles of fibers are little channels that CSF uses to go down into the nasal mucosum, where lots of lymphatic vessels will carry it away. Okay, so notice that it changes a little bit with age. We have a 26-year-old CT scan in the middle, and on the right-hand side is an 80-year-old. And just like the previous speaker discussed, your face changes with age, and these are parts of the bones of the face. Okay, I want to show you one more image, and this is a CT scan. It's right through, sort of horizontally through it. So you're looking at it from the side. On the top, the gray area is the olfactory bulb, and underneath that shiny white stuff, that's the cribriform plate and all the different features, because it forms conduits and apertures. And in between that, if you notice, there's a band of gray in between. That's the subarachnoid space. It has metabolite-laden CSF that's come all the way from the medial temporal lobe and the basal forebrain, and it has to get out of the brain. So the subarachnoid space forms little channels that go down into the apertures. It looks like this. So here's a cribriform plate, and in panel B there, we see the neurons shown in orange and these subarachnoid evaginations shown in blue. If we look to the panel to the right and see, that's just what the subarachnoid evaginations look like. And you compare that to an Alzheimer's subject in D, and they're pretty much gone. Okay, and one of the reasons they're gone is because the apertures are closing. Okay, so we did a study, which we're just publishing now, with hundreds of subjects, and we said, okay, where are all the apertures? And you look at the image on the left, you can see all the apertures. And the subjects under 55, that panel, the apertures, there's a heat map in the middle, the most frequent probability are at the very bottom, number 2, okay? And you can see the probability shifts from that to the panel to the right, people over the age of 65, all right? That data is shown on the far panel on the right, where you can see in the white bars, the number of apertures declines between people under 55 and people over 65. And people with Alzheimer's, which are the far right group, they have about the same, but the difference is their apertures are smaller. The cross-sectional area, the total area in people under 55 is the dark gray area. It's 7 versus 45 for people over 65, and half that for Alzheimer's subjects. So what's really happening is that this drainage system is slowly being choked off, and it's causing less fluid to go through and clear out that detritus, and it's accumulating, and it causes the formation of amyloid oligomers and then plaques and other inflammatory mediators that induce the formation of tangles. So it's really a plumbing issue. And the paper that I mentioned, it's under peer review, but you can see all this data on the top right in that QR code. Okay, so we have some very great data here showing that we have a decline in the flow capacity of the cribriform plate with age that's more advanced in subjects with Alzheimer's disease, but correlation does not imply causality. So the way we proved it was with an animal model. So we took some ferrets, and our human neurosurgeons made a little hole in the top of the snout, pulled the tissue off of the cribriform plate on the nasal side, and plugged the holes with dental cement. Okay, and we followed those animals for 7 months, and we tested them for spatial temporal memory, because that's one of the first things that goes in Alzheimer's disease. We used a tunnel maze for that. This is basically the same maze with two mirror images. We alternated left and right. If you look at the rightward maze, when they go through the tunnel, they're little green arrows. They would have to make 3 right turns to solve the maze. The next month, they'd be on the left one. They'd have to make 3 left turns. They run through it 5 times, and the 4th and 5th time, we get an idea of how good they are at spatial temporal memory. And what happened was the dashed line are the control subjects. So the control ferrets, they learned how to solve the maze. So their scores, their times are going down. But the solid line are the occluded ferrets, and their times are going up. They're having a progressive decline in spatiotemporal memory, right? And that's indicative of pathology in the medial temporal lobe. So we looked at the brains, and here we have a control ferret on the right. The olfactory bulb is at the very top. This is, again, looking from below. You can compare that to the occluded ferret on the left, and notice that we have significant losses of 40% of the olfactory bulb. Now look halfway down, and the green area highlights the medial temporal lobe equivalent for ferrets, and it's also reduced and smooth. It's 40% smaller, significant atrophy of this area. So this provides experimental evidence that we have a decline of cribriform plate porosity or flow with aging, with Alzheimer's disease. And if we block that experimentally, we get an Alzheimer's-like effect. Now, how do we translate that into something that's good for patients? Well, what we did was a Project CRIBROS with colleagues at Arrowhead Regional Medical Center. And what we did was recruit subjects, give them a CT scan, a 20-second CT scan, run them through some clinical analysis, some tests that I'll discuss in a minute, and a personal history. So it started off great. We had a dozen, then 2 dozen. We went to Alzheimer's walks. We recruited at senior centers. We recruited at bingo halls. We got up to dozens and hundreds, and we were doing very well, and then COVID hit. Fortunately, we had well over 500 subjects, which was enough for us to do some serious machine learning on this data. So this is what the scan looks like, a cone beam scan of a live subject, the cribriform plate. Now, it doesn't look like much. There are 2,000 gray levels in this, but we can only see about 20, okay? That's why we needed to create a deep learning 3D UNet model and train it to do that. So this is what that cribriform plate actually looks like when we process it with our system. We can see where the apertures are, and we can see the shapes, and that provides us an incredible amount of data on the morphology. We also developed something called the Leucadia memory test, and it's about a 15-20-minute test, and we tried to make it as engaging as possible. We used actually a video game engine to make it graphical. I've got a video to show you later of what it looks like, but it provides some tremendous data. So here we're plotting chronological age on the bottom, and we're plotting something called brain age, right? And this is like that clock you were talking about. The red line in the middle is the mean, and the green lines are 1 standard deviation away. So just about everybody fits in the middle. But at the very top, on the right-hand side, the people lined up at the top, they have a much older brain age than their chronological age. That's where subjects with Alzheimer's disease and other dementias cluster up there. And the people below that, they have mild cognitive impairment. Now, 20% of seniors have mild cognitive impairment, but not all of them progress to Alzheimer's disease. We think we can figure out who's who with our system. So we bring in our imaging data. We actually use the scans for it. Our Leucadia memory test, which evaluates 7 aspects of memory. We do an olfactory test, a 10-question smell test, and we ask personal history questions like, have you ever had a head injury or a broken nose, right? And so together, that allows us to provide something for subjects. So say this guy, Sam, is 68 years old, memory problems. He's got a family history of Alzheimer's disease. Doctor doesn't know what to do, gives him an MMSE, but that the results are unclear to him. His options are, 1, wait and see till dementia hits him like a freight train. 2, refer him for an expensive cognitive assessment if Sam happens to live in New York, LA, or Chicago, some big center. and it'll take a few months and be quite expensive. The third solution is to do ProCogny. So the evaluation that we've developed, it can be done in the office, on a laptop, on a tablet, on the phone. Sam can take the test at home every month on his phone, right? If we need a scan from Sam, the scanners we use, the cone beam scanners, are common in dental practices. So we can arrange for him to go to a dental practice, sit for 20 seconds, get a scan, comes back, and then we'll provide his doctor with a write-up telling him about what aspects of his cognition are failing, what it means, what the anatomical findings are. And at the bottom left there, we can actually plot out the rate of change for Sam and say, well, it's going to be, maybe 5 years before Sam goes from MCI to Alzheimer's disease. Okay, so one of the key features of this is that we have the memory test. So it was so engaging that we turned it into an app that can be used worldwide. So Leucadia memory test. So here, let me play you the ad.
Ever feel like your brain needs a jump start? ProCogny's got you covered with fun puzzles and games to exercise your memory, attention, and speed. Challenge yourself with premium puzzles and track your results. Then take a free memory test and see how you stack up to others your age. Download and play for free. Subscribe for ad-free play and access to premium assets. Get your brain on with ProCogny Memory Tracker. Yep.
Okay, so that's on the left there, the large icon. That's the ProCogny Memory Tracker. We think of it as like a Fitbit for the brain, right? People can play games every day, can take the memory test, and we can track and find out how they're doing and provide that information to them and their healthcare providers. The other apps are games, components of that, which are actually kind of fun to play. It's all available on the Google Store, on the Apple App Store, 22 languages, 174 countries, and it's free to download and free to play. If you want to play some premium stuff, it costs you $2 or $3 a month. Okay, so this is a way for us to do Alzheimer's research on a global scale. Okay, so let's go back to Sam. Let's say it's worst case scenario, occluded cribriform plate, MCI, headed for Alzheimer's. We'd like to provide a solution for him. And that is this, it's Arethusa. It's an implantable device that basically restores the clearance of CSF from the pocket right above the cribriform plate. So it's not really just a hole. There's a little more to it than that. This is from the approved patent. And it's accessible right up there. If you notice, this is an insertion device going up a nostril, but this is the route neurosurgeons use to operate on the pituitary gland, which is to the right and below. So it's very accessible for them. And in fact, our device has been put into 4 cadavers by our surgical team. And our head neurosurgeon, Dan Muley, says it takes about 5 minutes. He can put it in a patient in about 5 minutes. So this would be an outpatient procedure under twilight anesthesia, which is used for wisdom teeth, and the patient could be out and around. We think that this could be something like a pacemaker, but instead of keeping your heart going, it's keeping your brain going. So in summary, our products are like the memory track and the other apps. It provides us cognitive and memory testing for people all over the world. And then there's the clinical version of that with the scan. And that's a digital health tech solution that we could use with healthcare organizations like the VA or National Health Service in Britain to do large populations. Everybody over the age of 50 could start doing it, and we can plot the data and figure out where they're going to be in 5 years and what interventions are appropriate. And the last one is the brain drain, Arethusa, and it's a therapeutic device that we want to put into clinical trials in 2022 with people with mild cognitive impairment, probably some people from our study, from Project CRIBRIFORM, and it should improve their MCI and protect them from progressing to Alzheimer's disease. So we're also integrating with the incredible data you can get from smartwatches and cell phones. So my small smartphone has accelerometers, and we can model a gait for a person and find out if they have a Parkinsonian gait, do they have movements like Huntington's, do they have a stroke? This is all data that others are working on, but we'd like to bring it into ours, and we certainly have the resources and ability to do that. So it's surprising that this large amount of health tech data is the big players have sort of got away. Google Health lost their head guy and they're shutting it down. They still have Calico, DeepMind, and Verily, although how they'll play together is unclear. Haven from Amazon and JP Morgan and Berkshire Hathaway, they shut that down last year. The only one remaining is Apple Health. They've got the devices, they've got the data, years of data. And the great thing about it is little companies like us, we can ask our participants to grant us access to their data and analyze it and include it in our evaluation. So that's kind of it. This is our team. I'd like to thank everybody on our team, of course. And it's been great. Happy to take any questions. Okay, have to stop sharing.
David, you're on, or Dennis, you're on mute.
Yes.
There you go.
Thanks, Doug. What a big problem. Why do you think that, well, firstly, other than the publication you have coming out or preparing right now, are there other publications that support this thesis?
Well, this is the idea that the CRIBRIFORM plate is central, an apical role in Alzheimer's disease. I originally published in 2014. And in that time, a tremendous amount of data has come. People have been hunting for cerebrospinal fluid, and a lot of papers have come out on CSF playing a key role in brain health. Now, many of those studies involve something called lymphatics, where they found lymphatic-like vessels in certain areas, and it's debatable about whether it's inside the brain or outside because the arachnoid layer is the dividing line, and it's in that structure. But the CRIBRIFORM plate, it's well known, but it wasn't known how it worked, and so we really filled in those holes, and we're pioneering in this way. And the interesting thing is, I've been doing Alzheimer's research for 25 years, and we did cell models and mouse models, and my lab at the University of California, we did it there, and we ran clinical studies. And what I came to before I published that study was to sit down and say, well, listen, we're not making any progress. It's not like we're moving the ball down the field a foot at a time at all. It was 10 years of nothing. The only thing around were these anti-A beta studies, which have a tremendous amount of money and power behind them, but they don't work, right? And because what they're doing is they're targeting an effect, and we were interested in the cause. So we're not saying that once you have Alzheimer's, you do this and you'll be great. That's not the case. We're saying that you stop it from the start and you'll be in great shape. So there's a lot of data now about CSF and brain health, and it's becoming widely accepted. But here we're providing the mechanism for the clearance from one specific area. So
is your shunt going to be regulated as a device?
Yes, it will be a device. And as you, I'm sure, know, the devices, the bar for regulatory approval of devices is different than it is for pharmaceuticals. So what we want to do is initially a 20-subject study to establish that the device is not dangerous, it's accessible, it's easily removed, and that it provides efficacy for the subjects. And we should see beneficial effects in a month or two. So people with MCI, kind of foggy, once the device is in, it'll clear away the detritus and their memory should improve, and it should keep improving compared to a cohort that does not get the device, and they'll just progressively get worse. And so since it's so quick for us to get that primary endpoint, we think a 2-year study will probably be sufficient, because we'll put it into the subjects. It might take a year to put it into 20 subjects, and we'll follow them for a year. And at that point, we'll have quite a bit of data. And the great thing about that is we can continue to follow them for 5 years and then say, okay, our secondary endpoint, how many of them progress to Alzheimer's disease? Theoretically should be 0, versus the cohort where most of them would have progressed at that point. Right. So that's pretty much our game plan for the clinical trial.
Yeah.
Does this also work? You think it's going to be relevant for other neurodegenerative diseases?
I think it's very relevant. Very similar disease is frontotemporal dementia.
Okay.
Now it's similar to Alzheimer's disease, much more aggressive, and it's much younger people. And we think that it's probably a very similar mechanism at work there. And the thing is, as I said, these are facial characteristics. So I think there's a predisposition based on the size and shape of your cribriform plate, which you inherit from your parents, which probably is why there's a polygenic component to Alzheimer's disease. It's been unclear. I think one of the interesting things for us is COVID, right? Because as I may not have mentioned, an early thing that always happens in Alzheimer's disease is the loss of smell. And that's what happens in COVID, probably due to pathology in and around the cribriform plate. Now, people that have long COVID, they're slow recovering and they have sort of a brain fog. And it'd be very interesting for us to do a study and find out what's going on with that. Because if it causes injury and the repair causes some type of scarring that disrupts this system, a proportion of those people will progress to more mild cognitive impairment, eventually Alzheimer's disease. It would be nice to know that now instead of 10 years from now. So.
Now, as I understand it, Parkinson's also has a loss of smell, loss of taste, and just normal aging tends to have that too. So you'll be able to make that distinction from the tests?
Well, absolutely. We do the cognitive tests. You're absolutely right. There is a decline in olfaction with age, when Alzheimer's is a little more extreme. And the question is it cause or effect? Is it because everybody's going this direction, but some people a little faster than others, right? People lose their smell, or some people have their smell till they're 95 or 105 because of the shape of the cribriform plate that they inherited from their ancestors, right? The size of the apertures, the distribution. Also, life events impact this, because head injuries are associated, traumatic head injuries are associated with a higher risk for Alzheimer's disease. And many of those injuries involve a broken nose. So the middle of the cribriform plate, that little structure that aligns up, it's connected directly to the perpendicular bone in the nose, which is the cartilage in front connects to that. So if you deviate that, somebody falls off the bike when they're a kid, it deviates. And many of the subjects in our study have a deviation of that. Sometimes it's minor, sometimes it's extreme, in which case the ossification on one side is different than the other, predisposing them. And that's
Doug, this is fascinating. I wish we could talk about this for many more hours even, and maybe we'll have a chance to take it offline. Honestly, we can carry on the conversation at the Catalyst Institute for those who are interested.